Hello students, hope you all are fine and doing well. Welcome to another lecture of Microbial Genetics. You are listening to your very favorite Sandeepan Sengupta, who is an assistant professor in microbiology at the Kingston College of Science. In our previous classes and lectures of Microbial Genetics, we have discussed about mutation, different subtypes of mutation like missense, nonsense and silent mutation, several other interesting subtypes of mutation like directed or adaptive mutation, hypermutation, about the spontaneous mutation and its prevalent mechanisms. As I remember, in my previous lecture, I had been discussing all about the molecular basis of spontaneous mutations, about the most prevalent mechanisms which bring or which lead to spontaneous mutations occurring naturally in all living cells. So, what are those mechanisms? Those mechanisms include lesions in DNA, tautomeric shifts, formation of AP sites, that is apurinic sites as a result of DNA lesions, and frame shifts leading to micro insertions and micro deletions. Today's topic of discussion is chemical mutagens. But to inform you in the very beginning that chemical mutagens is a very huge topic covering many different classes of chemical mutagens. Altogether, there are 5 to 6 different classes of chemicals which together act as chemical mutagens and interact with DNA to induce mutation. So, the objective of my today's talk or this talk is to cover, for your convenience, only 2 major classes of chemical mutagens for today, that is base analogues and alkylating agents. So after listening to this talk, I think you all would be able to answer to the very common questions related to the base analogues and alkylating agents, like what are those base analogues, what are the structural features of base analogues, how do they incorporate in DNA, that is as normal bases in DNA, and how do they lead to replication, and of course you will be able to answer many more questions. Now let us begin our discussion about chemical mutagens. So as you all know that mutation may be spontaneous or induced. So just now I spoke about the prevalent mechanisms which are involved in generating spontaneous mutations. Now what about induced mutations? What are induced mutations? Mutations which do not occur spontaneously in cells but are induced by certain agents are induced mutations. So what are the agents which are known to, classes of agents which are known to induce mutations? Altogether there are 3 classes of agents which can induce mutations: chemical mutagens, physical mutagens, and biological mutagens. The chemical mutagens include a variety of chemicals which react with DNA. The physical mutagens basically include 2 different types of radiations, that is classes of radiations, ionizing and non-ionizing radiations. And what are biological mutagens? Hope you have heard about them, though I'll be discussing about it in my further classes. The biological mutagens are nothing but the transposons which are really wonders. They act as moving generic elements and they are capable of transposing from one genome or one chromosome to another. Now let us move on with the topics of today's class, that is chemical mutagens. So chemical mutagens include a variety of chemicals which interact with DNA and which act as normal, that is have a property of getting inserted as normal nitrogen bases in place of normal nitrogen bases in DNA and thereby cause mutations. So the first class of chemical mutagens are the base analogues. So what do you mean by base analogues and what are these base analogues? Base analogues are actually the chemicals which resemble DNA, purine and pyrimidine nitrogen bases in terms of their structure, but will show faulty base pairing properties. So the base analogues show structural features and have a structure very similar to that of one of the four nitrogen bases of DNA, that is adenine, guanine, cytosine and thymine. But they will show how they will induce mutation when, as a result of this, they get incorporated into DNA. But when on being incorporated, they will show faulty base pairing properties, and in turn they will form erroneous pairs, base pairs, leading to mutation. The two most, let us take an example and get into the more detail of the that is base analogues. Now, the two most common base analogues are 5-bromouracil, which acts as a substitute for thymine, and 2-aminopurine, which acts as a substitute for adenine. Now see, it's very interesting to look into the matter that how does this 5-bromouracil induce mutations in DNA. 5-bromouracil has a structure and which is almost similar to that of thymine, and 2-aminopurine has a structure which is almost similar as to that of adenine. So when incorporated, they can incorporate themselves as normal nitrogen bases in DNA. So when incorporated, replication error occurs normally most of the time. But when that is a base analog instead of a normal nitrogen base is incorporated into a DNA, the frequency of replication error increases at such a site. So what happens is that incorporation of the wrong base generates replication errors because it causes faulty base pairing and hence in turn induces mutation, which is detected during subsequent rounds of replication and segregation during the segregation process. Now, what happens with every base analog, say 5-bromouracil or 2-aminopurine? Say 5-bromouracil, it will normally assimilate, and instead of a normal nitrogen base, acting as a substitute for thymine. So it assimilates normally, but what happens is that in course of replication, it again the phenomenon remains the same, and what comes into play over here is tautomerism. That is, as a result of tautomerism, which has been discussed in very detail in my previous classes, the base, that is analog 5-bromouracil, will take an unusual or a rare tautomeric form with which it normally exists in chemical equilibrium. So the normal form of 5-bromouracil is a keto form, but when incorporated into DNA, as a result of tautomeric shift, it will change its structure and it will adopt another structural form which is called enol form. So 5-bromouracil enol form, when 5-bromouracil adopts the tautomeric enol form, instead of pairing with it is supposed to pair with adenine because it is a substitute for thymine. The keto form of 5-bromouracil can pair with adenine. However, when it changes to the unstable tautomeric form, that is the enol form, instead of adenine, it is not able to pair, does not pair with adenine, but instead pairs with another purine, that is 9-membered ring base, that is the guanine. So what happens in course of the process? Now this generates a faulty base pair, the AT base pair in course of subsequent rounds of replication is being replaced by GC base pair, which is a kind of transition base pair substitution mutation. I hope it's very clear to you. Now it will be more clear if we consider one more example, that is 2-aminopurine. Same is the thing with 2-aminopurine, and the one which plays the major catastrophic role over here is again the phenomenon called tautomerism. So 2-aminopurine is a substitute for adenine and it incorporates into DNA bases in place of adenine normally. But its usual form is amino form. However, in course of the replication process and after incorporation, it adopts a rare tautomeric form which is called the imino form of 2-aminopurine. So when it adopts a tautomeric form that is unstable or that is rare, the rare tautomeric form of 2-aminopurine shows quality base pairing properties with altered hydrogen bonding properties. So what happens is it actually 2-aminopurine and our adenine as such is supposed to pair with thymine, but the imino form or the tautomeric form of 2-aminopurine instead of thymine will pair with cytosine, another pyrimidine. Hence, in course of this process, it causes, or the AT pair is replaced by GC pair, which is again a type of base pair substitution called transition. So what happens is that every base analog shifts into a tautomer, incorporates into DNA, shifts into or adopts a rare or unstable tautomeric form and shows faulty base pairing properties, and in turn it can lead to or leads to base pair substitutions. So this is how the base analogues work. Now I have a very interesting question for you all as a part of your activity. Now just keep on thinking. I hope you are in a state to come up with the answer to this question. Think about that. Think about this question, that why does only that do you think that ribose can act as a base analog? What should be the answer to this? If yes, then why? Or if it is no, if not, if it cannot act as a base analog, then why? Keep on thinking about this. So I'm going for a very short break and I'll resume back after the break. So see you all after the break.